Hi everyone, today we're going to learn about enzymes. Topics include the enzyme substrate complex, the effects of temperature and pH changes, cofactors, competitive and non-competitive inhibitors, denaturation, the induced fit model, activation energy, optimization, and the difference between catabolic and anabolic reactions. We'll begin by talking about enzymes and the induced fit model. This is an enzyme, a catalytic protein that speeds up a reaction by either breaking or building up a substrate, which is the reactant that an enzyme reacts upon. The interaction between a substrate and an enzyme is demonstrated in the induced fit model. To start, a specific substrate moves towards an enzyme. The substrate moves towards the enzyme's active site, the region on the enzyme where the substrate bonds. The active site of an enzyme is flexible, so once the substrate is near it, the active site slightly changes to conform and mold to the specific shape of the substrate. They have a complementary shape to one another, and it is held by weak hydrogen bonds. Thus, we create our induced fit model, where they fit pretty close but not exact. The pair is now an enzyme-substrate complex. Let's talk about products. After the substrate is held with the weak hydrogen bonds, the enzyme converts the substrate into products, and the products are released into the reaction. This is called a catabolic reaction because the one substrate was broken down into two products. And then we see the enzyme opening up to receive new substrates because the active site is flexible. The opposite happens in an anabolic reaction, where two substrates are built up to form one new product. And again, the enzyme opens up for its active site to receive new substrates. Now let's talk about cofactors. Any non-protein molecule or ion that is required for the proper functioning of an enzyme. They can either be permanently bound to the active site or may bind loosely along with the substrate during catalysis. Cofactor molecules might be inorganic, like a metal such as zinc or iron, or an organic molecule, which is called a coenzyme. In this example, let's pretend that the active site of an enzyme is modified, so that it's no longer able to accommodate for the same substrate. The enzyme isn't able to perform catalysis anymore, so the cofactor comes in and provides the structure needed for the substrate to bind to it, creating that same induced fit. Cofactors might be helpful, but inhibitors aren't. They reduce the productivity of the enzyme. The inhibitor gets to the active site of the enzyme, preventing the substrate from binding to it properly. In this case, the inhibitor competes with the substrate for the same spot in the active site, making this a competitive inhibitor. A different type of inhibitor binds to the enzyme away from the active site. The substrate is still able to bind, but the inhibitor prevents proper function because it causes the enzyme to change shape in a way that makes the active site less effective at catalyzing the substrate. This is referred to as a non-competitive inhibitor.
Now we're going to talk about activation energy. In this graph, you can see that on the left side, we have the reactant, and on the right side, we have the product. At the top is called the transition state. This is a point in the reaction where the reactants start to transition into products. At the top of the curve is the activation energy without an enzyme. And this dotted line that you can see, this new curve, is the activation energy with an enzyme. What these two curves represent, or rather what the dotted curve represents, is that a reaction that takes place with an enzyme lowers the activation energy. In other words, the reaction requires less energy to take place because of the assistance of the enzyme. This occurs when a substrate binds to the active site of the enzyme. It is important to note that enzymes can help the forward and backward reaction processes. Next, let's talk about denaturation. Denaturation is when an enzyme can no longer function properly. This happens if an enzyme changes shape. So here we have an enzyme. One way that an enzyme can be denatured is an increase of temperature. As the temperature goes up, it causes the enzyme to change its shape, which ultimately leads to it not being able to function or no longer being active. A second way that an enzyme can denature is a change in pH levels. This can happen if an enzyme is taken out of its pH and into a higher pH environment or a lower pH environment. This example, we have an enzyme in a pH environment of 4 and is put into an environment with a pH of 10. This causes the enzyme to lose its shape and denature, rendering it inactive. Now let's talk about optimization. Each enzyme has an optimal temperature and pH at which its reaction rate is the greatest. In this first graph, you can see the rate of reaction versus the temperature. And for this particular enzyme, which is just a random one, the optimal temperature is at the peak of this curve. When the temperature gets too high, all the way to the right of the curve, the enzyme is denatured. On the left of the curve, near the origin, this is when it requires kinetic energy in order to get to the top of the bump. Something similar can be seen when we look at the rate of reaction versus pH graph. Same type of curve, and at the top of the curve, at the top of the hump, there is the optimal pH for this particular enzyme. When the pH gets too high, all the way to the right of the curve, the enzyme is denatured. However, what's different is that all the way at the left of the curve, near the origin, the enzyme is also denatured because it is too low. This tells us that every enzyme has a particular pH range that is specific to that enzyme in which it can exist in without being denatured.
Thank you for watching. We hope you learned something about enzymes. Here are the resources we used in case you wanted even more information. This video is brought to you by Chelsea Mills and Julia Weiss. Thank you again for listening.